Hi, welcome back to Bioinformatics Introduction and Methods. Let's continue with our case study. In this final unit of this week, I will show you how we use bioinformatic analysis of high-throughput data to infer the origination of de novo genes. This work was done by two talented former graduate students of mine, Chen Xie and Yong Zhang, in collaboration with another talented former student of mine, Chuanyun Li, who is now a faculty member at the Institute of Molecular Medicine at Peking University. Chen is going to University of Chicago to do a postdoc, and Yang is currently a principal investigator at the Institute of Zoology at the Chinese Academy of Sciences. So after we discovered a human-specific de novo protein coding gene, FLJ33706, we wondered how many other human-specific de novo proteins are there, and where did they originate from? To study these questions, we first had to develop a bioinformatics pipeline for the genome-wide identification of human-specific de novo protein coding genes. Here is the pipeline that we had developed. We also identified de novo protein coding genes that are specific for human and chimpanzee, or specific for human, chimpanzee, and orangutan. I will show you the pipeline for identifying human-specific de novo protein coding genes here. Most of the data and software we used come from the public bioinformatics resources that I showed you earlier, such as the genomic sequences, ensemble annotations, BLAST, etc. The pipeline has three main steps. The first step assigns the origination time of the genomic locus. We followed the method published by Yong Zhang, Man Yuanlong, and colleagues in 2010 in PLOS Biology. The origination time of the locus was determined by scanning different genome sequences for the presence or absence of genomic DNA segment homologous to each of the human gene loci. The origination time was set to be the divergence point between the group of species that have the genomic locus and the closest species that doesn't have the genomic locus. In the second step, we assigned the age of the open reading frame. For each locus in each outgroup species, an open reading frame is considered absent if two conditions are both satisfied. First, reliable codon-based alignment, that is, alignment with greater or equal to 70% coverage and greater than or equal to 50% identity, shows that the maximum continuous peptide before the first open reading frame disabler was shorter than 70% of the human ORF. Second, ensemble annotation did not identify any ortholog. In the third step of our pipeline, we determined whether the human gene is a de novo protein coding gene. A human gene is considered de novo if the following conditions are met. First, to ensure that it is a high-quality protein coding gene in human, it needs to have an intact ORF with RNA-Seq RPKM score larger than 0.5 in at least one of the 9 human tissues. Standard start and stop codons and intron lengths no less than 18 nucleotides. Second, it needs to have at least one unique supporting peptide from mass spec data in Peptide Atlas or PRIDE. Thirdly, we require that BLAST-P and ensemble found no homologous proteins in other species and no paralogous proteins in human, using E-value cutoff of 10 to the minus 6. Fourthly, the outgroup species should have no intact ORF. We discarded cases where the stop codon containing exon was spliced out in rhesus macaque. Finally, we require that multiple outgroups share a common disabler. This last criteria is important for distinguishing origination of new gene in human from loss of an ancestral gene in the outgroup species. Here is the example of FLJ 33706 again. How can you be sure that it is the acquisition of nucleotide G here that generated a complete open reading frame? Couldn't it be that the ancestral species also had the G and therefore a complete open reading frame, but it was lost in chimpanzee, gorilla, orangutan, and rhesus monkeys? We distinguish these two possibilities by the parsimony rule. Here, chimpanzee, gorilla, orangutan, and rhesus monkeys all have the same nucleotide A, which resulted in a stop codon. It is highly unlikely that all these 4 primates independently had the same G to A mutation. It is a lot more likely that the ancestral nucleotide was A and human had an A to G mutation. Thus, it is a lot more likely that human acquired a new open reading frame here. Using our pipeline, we identified 11 human-specific de novo protein coding genes, 7 human and chimpanzee-specific de novo protein coding genes, and 6 human, chimpanzee, and orangutan-specific de novo protein coding genes. All of them do not encode proteins in rhesus monkeys and other outgroup species. The median length of these gene products in human is 150 amino acids, which is statistically significantly smaller than the lengths of all human protein genes. 18 of the hominoid-specific genes have single coding exons in human, whereas most human genes have multiple exons. Interestingly, similar to the case of FLJ 33706, allele elements contributed to the creation of exons in 8 genes and the creation of splicing sites in 2 genes, highlighting again the importance of repetitive elements in the creation of new genes. We compared the expression level of the de novo new genes using RNA-Seq and found that it is generally lower than that of other protein coding genes in human. The next question we wondered was how these new genes acquired promoter regions. Using data from the UCSC Genome Browser, we found that 19 of the 24 de novo genes co-opted nearby transcriptional context, such as using promoters from an antisense transcript or using a nearby bidirectional promoters. Now, let's look at the question of how the hominoid-specific de novo protein coding gene originated from ancestral non-coding DNAs. There are at least 2 possibilities. The DNA sequence of the complete open reading frame may have appeared before it was transcribed and translated, or a non-coding DNA sequence could become transcribed first as a non-coding RNA, which could later acquire an open reading frame and became translated. To distinguish between these 2 alternatives, we integrated and analyzed RNA-Seq data from 19 tissues from human, chimpanzee, and rhesus monkeys. We found that 20 out of the 24 hominoid-specific de novo protein coding genes exist as non-coding RNA in outgroup species. So it seems that the majority of the de novo new genes originated from non-coding RNAs. Next, we asked whether the open reading frame appeared first or the regulated transcription appeared first. Here, we need to distinguish between 2 possibilities. One possibility is that the non-coding RNAs were upregulated transcription leakage or noise until the open reading frame was acquired and became regulated. Another possibility is that the non-coding RNAs already have highly regulated transcription profile and gene structure before the open reading frame originated. To distinguish between these 2 possibilities, we first looked at the RNA-Seq data in human, chimpanzee, and rhesus monkeys carefully to infer the gene structures. we found that the non-coding genes tend to have similar gene structure with their protein coding orthologs, including similar exon-intron structures and similar splicing junctions. Then we looked at their expression levels across different tissues and found that non-coding genes tend to have similar tissue expression profile as their protein coding orthologs. When we looked at the overall transcription level, we found that non-coding genes tend to have correlated transcription level as their protein coding orthologs, even though their transcription level is generally lower. So, which tissue do you think these de novo genes are most likely expressed in? It turned out that de novo genes have enriched expression in brain and testes. Similar to FLJ 33706, the isoelectric point of de novo new proteins are significantly higher than that of the whole set of human proteins. You may remember that we have previously found out that proteins with high isoelectric point tend to be involved in RNA functions. That's all we want to tell you about in this week's lectures. To sum it up, I hope we have convinced you that bioinformatic methods and analysis can play key roles in evolutionary biology by identifying interesting novel candidates at genome scale, discovering genome-wide patterns, and discovering cross-species patterns. Thank you for being with us for this week's lectures. Bye-bye.